Okay, before we start, do you have any questions about the homework or the structural alignment techniques in general? Today, the plan is to see the details, to talk about the details of another structural alignment technique, DALI, which is based on a different way compared to structal, which is based on dynamic programming, iterative dynamic programming. DALI is a different heuristic which tries to find the similarities between protein structures. So this is a reminder of the recap of what we have been talking about last week. This is an example which structal runs. Okay, so what we were doing in structal, starting with an initial arbitrary alignment of amino acids, matching of amino acids. We use superimposition. The superimposition technique is used iteratively to find whether these matches can be changed, updated, based on how close these amino acids are in 3D space. So the first superimposition gives us something like this. Based on the arbitrary alignment, we get something which is not correct, not a very good alignment. The superimposition does not reveal the similarities between two structures. I mean, if you just look at these two structures, you can see that there is a linear portion, and that linear portion matches the linear portion here in the second structure, although there's a kink here in the second structure. These 5 amino acids, they also form a linear shape, which is the same shape here. So they need to be matched with each other for the correct similarity match. But the first superimposition, as you can see, fails to find this structural similarity, but after that, after the first superimposition, we can change our initial alignment by matching new amino acids with each other based on how close they are in 3D space. So we construct a scoring matrix based on how close amino acids are in 3D space, and this scoring matrix is used to run a dynamic programming algorithm, the same sequence alignment algorithm. Only one thing that is different from regular sequence alignment is that we do not use sequence matching score. Instead, we use this scoring matrix we have derived for three-dimensional positions. So we find new matchings, and based on these new matches, we re-superimpose. So this iterates until your amino acids that are matched do not change. At that point, your algorithm converges to some local optimum. Okay, so now, before we start on the DALI method, let's try to think about how we would, I mean, if I ask you to think about an algorithm to match structures. So if I give you 2 protein structures, or if I have you solve this problem. I have 100 protein structures in a database. I have hundreds of them. And I give you one query protein structure, and I ask you, find the one which is most similar to this, find the protein structure in this database of 100 protein structures, which is most similar to this new one I gave you. So what would you do? And when I say protein structures, you should think about what you have in terms of data, is that you have their sequences, amino acid sequences. You also have atomic positions, 3-dimensional coordinates, x, y, z coordinates for each of these amino acids. And you may imagine each amino acid is represented by just one point, one coordinate, which is the coordinate of its alpha carbon atom. Okay, and these points are in 3-dimensional space. Okay, they are in x, y, z coordinates. If I tell you how would you solve this problem, how would you find which structures are similar to the other structure? I mean, try to think about some, even if you're not a computer scientist, you may think about some automatization techniques, or how would you approach this problem in the first place? Before starting to write a program to do this, how would you approach this problem? So you would do sequence alignment to find whether you have a similar sequence. And what happens if the highest similarity sequence you have from the database just contains 10% sequence identity? I mean, in other words, in terms of sequence, they have no similarity. It turns out that in this specific example, sequence similarity does not lead you anywhere. So how would you compare the structures? What would you do? Yeah, even maybe less than that. I mean, sequence similarity does not tell us anything about similarities.
I use the best one.
Okay.
I pinpoint match the exact matches as the exact matches.
You superimpose them. You superimpose them on top of each other, and see whether it gives you a similarity. Okay, in that case, it may be the case that a 7% sequence identity structure sequence may have a much higher similarity than what you have found. I mean, you may miss the actual, your solution may give you an incorrect result. The actual structural similar protein may have maybe 7% sequence identity. And if you look at 3D shapes, you see that they are really similar. So the question is, how do you find these really structural similarities? I mean, just think about if you didn't have any programs, what would you do? You would just manually look at their pictures, right? I mean, like if you have a visualizer, you just plot these points on 3D space and look at their shapes manually to see in terms of shape, there are certain similarities. So as humans, what do we do manually? When you look at these shapes, how do we decide whether two shapes are similar or not, for example? And maybe if we think about this, we can maybe convert our thought process into a program to find the actual, to find this automatically. Yeah, so shapes. So how can you, I'm trying to get to this, this thing, which is, if two shapes are similar, what do they share? For example, their XYZ coordinates, are they going to be similar? Edges. Okay, so now we are getting somewhere. What do we mean by edges? The angles between, for example, the bond angles, you mean? So for here, what we are trying to do here at this step, you are getting to a process of extracting certain structural features. Okay, so what you do, an edge, the angle between two edges, is a structural feature. I mean, meaning that a property, a structural property. What other structural properties you can use? For example, are there XYZ coordinates, structural properties? Can you directly use them? Why not?
Because there may be different aspects, I mean, in different
coordinate reference frame. Maybe one of them, yeah, that was my point actually. If this is the origin, okay, 0, 0, 0, one protein may have been solved here. Another protein may have been solved in this region of the coordinate reference frame. So it may have all negative XYZ coordinates. This one have all positive XYZ coordinates. By just looking at XYZ coordinates, you cannot say that they have different shapes. Okay? I mean, the coordinate, just the coordinates itself does not give you anything about the actual shape. However, for example, can you compute the diameter of this shape? Finding the, what you are given, this shape, what you are given is a set of points actually. So you are given a set of points here, you are given another set of points here. All these points are in 3D space. So what other structural features we can use? Like diameter of this, diameter meaning the distance between the points which are farther away from each other. So the distance which are farthest away from each other is going to be the diameter. It will tell you some, I mean, diameter is a single number, a scalar. And it may tell you, it may help you to prune out certain very different shapes, but it cannot tell you anything itself, right? I mean, can you, because the structure, it contains a lot of information, this shape. If you look at it in terms of information content, the shape has a lot of things, okay? I mean, the angles, the volume it occupies, I mean, the shape as information content, shape contains a lot of things. If you try to summarize this by just single number, diameter, you will not be able to reconstruct the shape from a diameter. I mean what I'm trying to tell is I may give you exact same diameters for two completely different shapes. So diameter is not itself enough. So what we may do is we may add new features in addition to these diameters. What else can we add?
If we can denote atom like a point,
Mm-hmm.
if we can denote a shape as a center of geometry, maybe.
Okay, the whole shape we can represent this as the center of geometry, a single point like this. I have a single point like this for this as well. So are you going to look at the coordinates of these two single points to understand whether they are similar?
Coincide.
Okay, now you coincide, then what you do?
Then we change the shape. Conformation, change conformation.
Okay, so we make this center of gravity to be on top of this center of gravity. So we may have, so their center of gravities match each other. And then what do? We superimpose it. So superimpose by using which points? So for superimposition, we need to give matching points, saying that, I mean, how are you going to know you need to rotate it this much to get the best superimposition? So, yeah, point by point. So similar to structural, maybe we can find that, we may have an iterative algorithm here. So here, I mean, what you have described here is actually the same as the structural algorithm. I mean, just bring them closer to each other first, then try to find matching points. So what I'm trying to get is something different. Without superimposition, I give you two structural shapes. What can you measure about these shapes that is going to tell you about their shape? Something like volume is one example. The volume is one, just again a scalar numerical example. If you look at, for example, two proteins, if their volumes are very different, then their shapes are going to be very different as well. So probably in this database search, you can just prune out if your query protein has a volume of 100, for example, and the other, if there are certain proteins which are very small, their volumes are 10, you can just get rid of these proteins and saying that the structure is not going to be similar. Because we are not thinking about protein structural similarity, we are not thinking about scaling. I mean, their shapes may be similar. A smaller version is not considered a similar structure in protein structural space. We are treating them as rigid bodies. We are not trying to look for similar scaled down versions of the proteins. In terms of size, also they need to be similar. So what other structural features? So here, what we are looking for here is that, which is called a structural feature we are looking for here is something rotation and translation invariant features. I mentioned this like last week as well. What does it mean to have rotational and translation invariant features? If you rotate this structure, if you put this structure somewhere else, does its volume change? No, right? So volume is a rotation and translation invariant feature. So, and therefore, if we can extract such features, which are rotation translation invariant, it's a pure feature about its shape. It's going to be something about its shape. So our goal is to find such features. And any other ideas than volume and diameter? What can we, I mean, what numbers can we extract from this structure that is going to tell something about its shape? Yeah, distances, and actually this is what DALI uses. And it's this. DALI tries to do the same. What DALI algorithm tries to do is, it tries to represent the structure in another form, but by not using coordinates, because the original XYZ coordinates are not rotation and translation invariant. If you move this protein somewhere else, if you rotate it, its XYZ coordinates are going to change. Okay, so the XYZ coordinates by themselves are not rotation and translation invariant. But what about, for example, look at these two amino acids on this protein structure. And look at the distance between them. If I rotate this protein structure and change its rotation position, does this distance going to change? No, because it's a rigid body. We are not going to deform it. We are going to put it. So this distance is going to be fixed. If you compute this distance, d1, it's going to be a rotation and translation invariant feature, this distance. And what if you compute the distances between all pairs of amino acids in this structure? I mean, it's like you compute this distance. You compute all the pairwise distances between all amino acids within this structure. And this is actually going to be a lot of numbers. But what I'm going to tell you is that by using just these distances, if somebody gives you these distances, you can reconstruct the whole 3D shape. Whole positions can be reconstructed from these pairwise distances. So it tells you everything. I mean, it has all the structural information content, these distance matrices. And these are called intra-atomic distances or intra-amino acid distances. And DALI uses this structural feature. But this intra-atomic distance is not the only distance, not the only invariant feature we can extract. We can extract other features like what we can do is we can, for example, construct a three-dimensional molecular surface around these points. And we can, for example, measure certain curvature values, or I mean some geometrical, we can measure certain geometrical angles or curves. I mean, for example, we can look whether there are certain edges, there are certain hills that stick out from this protein. So we can compute other structural features that are rotation or translation invariant as well. So what I'm trying to say is this intra-atomic distances is not the only way to extract some features about the structure. That's just one way to do it. We can use other ways to extract structural features. Now, DALI is an acronym for distance matrix alignment. So what they do is these intra-atomic distances, since we are doing it between all pairs of atoms, what we have is for a protein structure can be represented as a matrix, as a symmetric matrix and square matrix. Okay, a protein structure is represented as a square matrix. We can turn this shape. We can turn these 3D coordinates into a square matrix in which the diagonals are all 0, 0, 0, 0. Why is it 0, 0? Because the distance of an amino acid to itself is 0. So here, an entry in this matrix shows the distance between amino acid I and amino acid J. So it's in 3D space. And since the distance between I and J is symmetric, if you measure that distance is a symmetric distance, we are going to get ourselves a symmetric distance matrix. And this distance matrix, if you compute this for real proteins, they look something like this. Okay? So this is I mean, what we are going to have, in a real distance matrix, we are going to have some numbers. I mean, the distance is a scalar number, like 3.5, 7.8, 10 point. I mean, they're real floating point numbers. Here, it is visualized by converting these distances by giving dark colors to small distances and light colors. So we can what we can do is we can linearly, so in this grayscale, 0 means black, 255 means white if we are using 8-bit colors, okay, for this for each color dimension. If we are using grayscale, what we can do is we can linearly normalize the values between min distance and max distance. For example, your minimum distance can be 0, and for example, your maximum distance is 300 angstroms, I'm just assuming. You can rescale these distances between 0 and 255 into this color space, and you are going to get, at the end, you can visualize your intra-atomic distance matrix like this. The diagonal is completely black, and why do we have not a narrow diagonal, but why do we have a diagonal like this, which is a larger band? What is the reason for that? I mean, the black region, okay, this is the black region. The black region looks thick. What is the reason for that? No. Just think about this. Amino acid I. What does the diagonal, for example, this is the exact diagonal, and these are diagonal neighbors, for example. If this is amino acid I, what is this amino acid? I plus 1. This is I plus 1, I minus 1, or I minus 1. Okay? And it's index in the sequence. How far can you be to I, if you're amino acid I, how far can you be to I plus 1? Not much, right? Because you're all bound together with a peptide bond. So we have a linear string of amino acids in the string. You cannot be very far away from your neighbors, left and right neighbors. That diagonal, the thickness of diagonal shows that. I mean, because each of these amino acids, they are tied to each other by peptide bonds in a linear way. You cannot be very far away from your neighbors in sequence. Okay, so you cannot be very far away from your neighbors, but so usually, I mean, this diagonal band, its thickness is not, it doesn't give us much information. I mean, it's based on the polypeptide bonding in the sequence. We expect a thick band like this. But you see some interesting structural shapes here. For example, you see certain off-diagonal lines like this. Look at this. We have these bands, which are perpendicular to the diagonals. For example, look at this one here, which is something perpendicular to the diagonal. We have a band here. What does that band, what is your guess about that band?
They are interacting.
Yes, exactly. So it shows, what it shows is that there are certain different parts of sequence. There are different parts of, this is your sequence. It shows that a certain part here is close in 3D space. It means dark regions means that they are close in, so black color is smaller distance. So if we have a band like that, it shows that these amino acids are closer to each other in 3D space to other amino acids somewhere else, other place in the sequence. So they're interacting. What kind of a secondary structure was that? Just remember secondary structures? Okay, is it a helix? No, it's not. It's because they're far from in the sequence, they probably form beta, they are part of a beta sheet. And is it a parallel or anti-parallel beta sheet? Yes, anti-parallel beta sheet, because of what it shows is that in one sequence, when the indices get higher, I plus 1, I plus 2, I plus 3. The other part is, its interacting residues are decreasing. So it's in reverse order. The interacting residues, look at this. Actually, this is shown, this matrix is shown in a, so here what we have, so it's 0, 0 starts from here. So as you can see, when one sequence we are increasing, for example, this band shows that the amino acids between 0 to around 30, so the amino acids between 0 to 30 are interacting with amino acids from 75 to 40, something like that. Okay. So, and here, the zeroth one, this dark region shows that the zeroth amino acid is interacting with 75. First amino acid is interacting with 74. So when in one sequence we are increasing 0, 1, 2, 3, 4, 5, 6, we are decreasing in the other sequence, 75, 74, 73. So it shows that we are in different sequence orders. So it's an anti-parallel beta sheet. And if we had seen a similar band, which was parallel to this diagonal, so if we had a band here, for example, a dark region, parallel to the diagonal, it would mean a parallel beta strand. What about helices? Can we see helices in this figure? Where are they going to show up? Yes?
If we see a black pattern,
Where should we see that black pattern? I mean, just tell me its position with respect to the diagonal. I mean, can I see a helix somewhere here? I mean, like a horizontal black pattern like this would mean a helix. Dashes mean, I mean, a horizontal black band, you mean. So let's see what it means. Let's try to see. In terms of distance matrices, so we have, for example, the ith amino acid. So we have this black region here, J plus 1, J plus 2, J plus 3, and goes something like that. So what you say is that there is a single amino acid, I. This band is going to represent, there's a single amino acid, I, which is close to lots of amino acids. J plus 1, J plus 2, J plus 3. And this is the ith amino acid. So does this what happens in helices? No, because there is no central amino acid in helices. What happens in helices is that the main contacts, so I mean, if you think about a helix, in addition to your neighbors in your sequence, so these are your neighbors in the sequence, what happens is that this one gets in close contact with this one, somewhere far away, and this one gets in, if we have the same rotation angle, it's going to get in touch with this one, but the first one is going to be far away the second one. So we cannot say there's a single amino acid which is going to be close to everybody in this helix. What we see here is that, for example, the ith amino acid is going to be close to the I plus 4th amino acid. I plus 4th is going to be close to the I plus 8 amino acid. So it's going to be something like that. And as you can see, these interactions, I and I plus 4, tells us that we cannot get very far away from the diagonal. I mean, when we are going to see these helical interactions, we are not going to see this helix, I mean this amino acid here, because it's helix, it's not going to interact with another sequence that is very far away in sequence. So in helices, we are still in close neighbors. I mean, I and I plus 4, I plus 4 and I plus 8. So the interactions of helix we are going to see are going to be close to diagonals. you cannot see in this figure very well, but certain places the diagonal band is thicker than in certain regions. For example, in this region it's thinner, but there is a certain thick region here. Maybe around, this region is a little bit thicker than the other parts. That thick region may indicate a helix, I and I plus 4, I plus 4, and I plus 8, and something like that. And anyway, what we see is, such a matrix can tell us a lot about shape. Okay? We converted something that was three-dimensional. A three-dimensional shape is converted to a 2D matrix like this. And what DALI does is, DALI processes these matrices and tries to find similar submatrices between two different. So if we are going to, remember, our goal is to compare the similarity of two shapes. So in order to compare the similarity of two shapes, we are going to have one matrix for one shape, the other matrix for the other shape. We are going to have their separate intra-atomic distance matrices. And the problem is reduced to comparison of matrices. And therefore, hence the name of DALI, distance matrix alignment. So it aligns, it tries to find similar regions between these intra-atomic distances. It's a fairly old method. The first DALI method was proposed in about 20 years ago. And they used it to find the similarities between all protein structures known up to now. So they tried to map the structural universe, protein structural universe in 1996. And they also have a DALI, there's a server which you can query pre-computed structural alignments. And here is, this, I mentioned about intramolecular distances or intra-atomic distances. So they extract, in terms of features, structural features, they extract these matrices. And the main idea is that if we place one matrix on top of another, if we find similarities between two intra-atomic distances, we are going to find similar shapes. That's the main idea. And the actual implementation is more complicated than this. To find similarities between two matrices of different size, so they try to form a matrix alignment. They break each matrix into smaller, I think in the original implementation it was 6 by 6 matrices. So each of these matrices are converted to 6 by 6 overlapping submatrices. And then first seed matches, submatrices of small size, and which very similar values are found between these two matrices. And then they try to combine these submatrices into a larger alignment. So based on similar idea as BLAST, right? This is what BLAST does. First, high scoring pairs, the seed matches, exact matches, and these exact matches are combined together to form the larger alignment or extended. In this case, these seed matches, the submatrices, can be very far away from each other, and they try to form a consistent chain of these submatrices to find the overall alignment. So the first way is we have a protein structure like this. The first way is to, the first step is to represent it like a distance matrix like this. And since the distance matrices are symmetric, what we can do is we can just use diagonal for one matrix and the other part for the other matrix. And the submatrices of fixed size are found first, and these are compared, and this pair list is high scoring pairs. They find matching, and this match is done just by comparing the numerical values of these 6 by 6 matrices. You compare two 6 by 6 matrices. If they contain similar values at each entries, you treat them as a contact pair or a similar substructure, and then it assembles these pairs in the correct order in a consistent way to find the overall alignment. And the assembly of alignments, assembly of these small sub-matrices onto a larger matrix is a difficult problem actually. It's a combinatorial problem, and they have different techniques implemented to solve that problem. One of them is based on Monte Carlo optimization, branch and bound search, and neighborhood walk. All of these are implemented in DALI. I'm not going to go into details, but I mean, this figure shows another way. It just shows an example of how similar contact patterns are grown to find a larger structure. For example, here we have two protein structures, okay, protein structure 1 and protein structure 2. Here, in protein structure 1, there are these two beta strands, A and B, that are close to each other in 3D space. And the second structure may have other beta strands which are also close to each other in 3D space. And these are going to be, these pink regions are going to be our matching, our submatrix. And this pink region, the other structure which is larger, as you can see, the second structure is larger. It has a larger loop region here. This one has short loops. So it also has this pink region. So the first step is to find that there's a pink submatrix like this, which is similar to this pink submatrix in the other one. And then they find B is close to C. B is also close to C in the other one. So this light blue regions are also similar here. Then what they do is they combine this pink and light blue regions in a consistent way to find that we have a substructure ABC which is similar to A prime, B prime, C prime, like this in the second structure. I'm going to skip these details, and I'm not going to talk about the lock algorithm. You can read this in the slides, but it represents the structure in a different way. The secondary structure elements, the helices and strands are represented as vectors. Then they try to first find an alignment between these vectors and then perform a more detailed atomic alignment. So, these are my last slides. VAST is another algorithm. Just you should only know their names, okay? In detail, you should know the structural algorithm in every step how it runs, the iterative superimposition, iterative dynamic programming technique. And DALI, you should know how we extract the distance matrices. What does intra-atomic distance matrices mean? How does it represent shape? And that's it. So that's all I'm going to talk about structural alignment. Do you have any questions? Let me talk about your fourth last assignment a bit. You are not going to implement any alignment technique, or you are going to be doing something we haven't seen in class. In your fourth assignment, it's going to be about structures. What you are going to be doing, the algorithm is going to be very simple. You are going to write a utility program which is able to parse a protein data bank file. My main reason of giving this homework is that for you to see actual structures. I mean, for you to show that, proteins are really, they have these atomic positions, they're 3D coordinates, X, Y, Z coordinates. So in the first part of your assignment, you are going to write a parser. Your parser is not going to be very difficult. You're just going to extract the X, Y, Z coordinates of alpha carbon atoms. From that PDB file, you are going to ignore a lot of information in that PDB file. You are going to only look at the lines which contains carbon alpha atoms and just extract the XYZ columns, XYZ coordinates. So at the end, as a result of your parse, you are going to have a protein structure represented as a list of XYZ coordinates. Okay, this is going to be your protein structure. After you parse it, it's a list of XYZ coordinates. What I'm going to have you do is, a protein structure, a quaternary protein structure, may contain multiple chains, right? Multiple polypeptide chains, it may be. So you'll also, actually after parsing, you're not going to get a single list of XYZ coordinates. For each different chain, for example, for chain A, you are going to have a list of XYZ coordinates. For another chain, chain B, you are going to have another list of XYZ coordinates. So you are going to extract these XYZ coordinates for multiple chains contained in the protein data bank file. And I'm going to, for testing, I'm going to give you protein structures which are composed of multiple chains. Okay, they are going to be, it's not going to be a single chain, and your goal is going to find out from these two, looking at these multiple chains, which amino acids of these chains are interacting with each other. In other words, this quaternary structure is formed by physical interaction of, I have one chain here, this is one chain, this is the another chain, for example. The protein structure is formed by interactions, by physical binding of one chain to the other chain. And your goal is to find out which amino acids are responsible at this binding. How are you going to do that? What you need is all you need to do is compute distances between all XYZ coordinates and all XYZ coordinates in this chain, and report the amino acids which have a smaller distance than a given threshold. And this threshold is also going to be user input. When the user says that report me all the amino acids between these two chains which are closer to each other than 3 angstroms, you are going to do that. You are going to report all the amino acid pairs in contact, meaning that they have smaller distances than 3.0 angstrom or 4.0, based on, because this contact definition is there is no universally accepted contact definition. Some authors use 3.3 angstroms as contact, some of them use 4 angstroms. So it's going to be a user input, and at the end, you'll have a utility program which can parse PDB files and extract interacting regions of these multiple chains. Okay, you'll have this very nice utility program that can do that. And as you can see, in terms of algorithm, there's nothing that's challenging. I mean, you just compute distances between pairs of points and report the ones which are smaller. Here, you'll have an experience to work with a real protein data bank file, see the contents, and maybe if you can do it, it will be good if you can visualize your result in some other external program. I mean, you may output your interacting amino acids position maybe as another PDB file, and you can view it as a separate, by using existing protein visualization programs, you can visualize the interacting region separately. You can look at the interacting region as a separate protein file if you can output your results also as a valid PDB file, for example. Yes? The PDB is a format. Yeah, the PDB has a known format. You can go to Protein Data Bank to learn more about the format, but if you just open some PDB files that I gave you, you are going to immediately see what the format is, and you are going to know how to extract this. You are going to ignore most of the lines. You're just going to look at the atomic coordinate. Yeah. There are certain tools actually that can find these interacting parts. I mean, but you are going to be writing it yourself. Okay? I mean, this interacting regions between two chains, it can be I'm sure this is a very important task that is for biologists to find these interacting residues, and therefore lots of utility, there may be existing utility programs that does this, but you'll be doing one for yourself. Okay. Well, that's it for today. So see you tomorrow at 10:40.